Chapter 9, Patterns of Inheritance. The question is why genetics matters. Every trait that an individual is born with results from inherited genes. Understanding genetics is therefore vital to such diverse fields as forensics, agriculture, and evolution. It also forms the basis for all inborn diseases that may affect a living organism. Now, if you look at the figure on the top left panel, you see some features are determined solely by genes. That means they are hereditary. Some by environment and some by both. Now, moving to the next figure, top right, every person's sex was determined by paternal genes, okay, from the father, the Y chromosome that is responsible. Now, looking at the figure in the bottom of the slide, your chances of inheriting and passing along a genetic disease can often be determined from your family history. Biology and Society, Darwin's dogs. Dogs are one of humankind's longest running genetic experiments. People have selected dogs for specific traits or characteristics and then bred them to create hybrids, and that is what has resulted in an incredible variety of body types and behaviors in present-day dogs. There are two important terms that you have to remember: genetics and heredity. What is heredity? Heredity is the process by which traits, that means characters or features, are transmitted from one generation to the next. So it is transmission of traits, while genetics is that branch of science which studies heredity, that means how the genes are transmitted. You see here the picture of Gregor Mendel, who is known as the father of genetics. Around 1860, Gregor Mendel found out that parents pass on specific traits or features in the form of discrete, that means separate, genes. At that time, the term gene had not been coined, so he called them heritable factors. So he proposed that parents pass on these heritable factors to their children, and these heritable factors or genes are responsible for the features or the characters that you see in the children. And then the genes or the heritable factors, they retain their individual identities generation after generation. Mendel's experiments were conducted on the garden pea plant. Scientific name for this plant is Pisum sativum. Mendel chose this specific plant probably because these plants were easy to grow. They had a very short generation time. That means from one generation to next generation, he didn't have to wait that long. And also the plant came in many different varieties which were easily distinguishable from each other. So let's say he wanted to study plant height. So he found that some pea plants were tall and some pea plants were short. So there were these easily distinguishable varieties. Now, two terms here are character and trait. A character is a heritable feature that varies among individuals, while trait is a variant of a character. For example, a character would be, let's say, the color of the flower. And a trait would be, is the flower white colored or is the flower purple colored? Or again, another example is a character would be the plant height, and the two traits would be tallness and shortness. So the characters that Mendel chose to study, the advantage was that these characters occurred in pairs. That means, let's say he chose the character of height. So pea plants showed some plants were tall and some were short. Or he chose the flower color. Some flowers were white colored, some flowers were purple colored, and so on. So he had a list of characters that he wanted to study, and each character existed in two alternate forms. And perhaps the most important advantage of pea plants as an experimental model was that Mendel could strictly control their reproduction. These pea plants could be self-pollinated as well as cross-pollinated. So he could do genetic experiments very well, and later on you will find that actually Mendel tried to reproduce these experiments in another plant model, and he couldn't do it. Okay, because those plants were asexual. So pea plant played a very important role in helping Mendel come up with his postulates or come up with his findings. This figure shows the structure of a pea flower, and the stamen is the male reproductive part of the flower, while the carpel is the female reproductive part. As I mentioned before, that pea plants could be self-pollinated as well as cross-pollinated. So whenever Mendel wanted the plants to be self-pollinated, he would bag the flowers. That means he would put a bag around the flower so that no cross-pollination could occur. And when he wanted cross-pollination, he would do it manually himself. Okay, he pollinated the plants by hands so that he was always sure of the parentage of his plants. And Mendel created in this way purebred varieties of plants and then started crossing two different purebred varieties. This slide illustrates Mendel's technique for cross-fertilizing or cross-pollinating pea plants. So he would remove the male reproductive parts, let's say from a plant that had purple flowers, and then he would transfer pollen from the stamens of a white flower to the carpel of a purple flower. So he was taking the pollen from a white flowered plant to a purple flowered plant, and then he would let, after the fertilization, he would let the carpel mature into pods, and then he would plant the seeds from the pod to see what kind of flowers the next generation of plants, which is the offspring or the F1 generation, F1 stands for first filial generation, what kind of flowers these plants had. In this slide, we are learning the basic terminology for Mendelian genetics. So what Mendel was creating was basically a hybrid. Hybrids means they are offsprings of two different purebred varieties. So because he took the stamen from a white flower and was pollinating the flower, the purple flowered plant, so that cross-fertilization process is known as the genetic cross. The two purebred parental plants are the P or the parent generation. The first hybrid offspring that is formed is the F1 generation. Now, if cross occurs between F1 plants, then the next generation becomes the F2 generation. Mendel conducted several experiments in which he was tracking the inheritance of characters, for example, the height of a plant or the flower color, and each character occurred in two alternative traits. And when he pooled the results from all of these characters, the results, the findings led him to formulate several hypotheses about how inheritance occurs. This slide gives you the list of 7 characters that were studied by Mendel in pea plants and their two alternate forms in which the character exists. For example, flower color is a character, and the flower color could be purple or white. Those are the two alternate traits. Or if you go to the right side bottom, stem length is a character, whether the plant is tall or dwarf. These are the two alternate traits. Mendel's first law is known as the law of segregation, and it deals with the inheritance of a single character. The next slide has a figure 9.5, which shows a genetic cross between a purebred pea plant with purple flowers and a purebred pea plant with white flowers. Now, when you see the next slide, you will find that all the plants in the F1 generation, the first hybrid generation, had purple flowers. However, Mendel realized or figured out that the gene for white flowers did not totally disappear in the F1 plants, but the genes were somehow kept hidden or masked when the purple flower factor was present, and that's why all the plants had purple flowers only in the F1 generation. He also deduced that the F1 plants must have carried two factors for this particular flower color character. One factor for purple and one for white. So from these results and others, Mendel developed 4 hypotheses. This is figure 9.5, which shows the inheritance of a single character and the law of segregation. You see, in P generation, which is parental generation, there was purebred plants with purple flowers and purebred plants with white flowers, which were crossed. In the F1 generation, all the plants had purple flowers. Then Mendel did a fertilization among the F1 plants. That means an F1 was crossed with another F1. And in the F2 generation, he saw a re-emergence of plants with white flowers which had totally disappeared in the F1 generation. Note the 3:1 ratio of the purple flowers to white flowers in the F2 generation. So the four hypotheses made by Mendel were, the first one is, there are alternative versions of genes that account for variations in inherited characters, and these alternative versions of genes are called alleles. Second, for each inherited character, an organism inherits two alleles, one from each parent. Now, an organism that has two identical alleles for a gene is said to be homozygous for that gene. An organism that has two different alleles for a gene is said to be heterozygous for that gene. A common example is in case of human beings. If you look at the sex chromosomes, in females it is XX. So the females are homozygous for the sex chromosomes in case of human beings. While in case of males it is XY. So males are heterozygous for the sex chromosomes. Hypothesis number 3. If an organism has two different alleles for a gene, then one allele determines the organism's appearance and is called the dominant allele. The other one has no noticeable effect on the organism's appearance and is called the recessive allele. So the dominant allele, that means dominates over the effect of the recessive allele. Now in genetics, we usually by convention use uppercase italic letters such as a capital P, that means an uppercase P, to represent the dominant alleles, and lowercase italic letters such as a small p or a lowercase p to represent the recessive alleles. Hypothesis number 4. A sperm or an egg carries only one allele for each inherited character because the two alleles for a character segregate, means separate from each other during the production of gametes. This statement is called the law of segregation because the alleles are segregating or separating. When the sperm and the egg unite at the time of fertilization, each contributes its alleles, restoring the paired condition in the offspring. For example, in case of human beings, we get 23 chromosomes from mom, 23 chromosomes from dad. That means 23 maternal chromosomes, 23 paternal chromosomes. Half of the genes are coming from the mother's side, half of the genes are coming from the father's side, and then they unite at the time of fertilization to restore the number of chromosomes, that is 23 plus 23, 46. And the genes again get back in the paired condition. In the next slide, you will see a figure which illustrates Mendel's law of segregation, and it explains the inheritance pattern that was shown in the previous figure. This figure explains the law of segregation. Mendel's hypothesis predict that when alleles segregate during gamete formation in the F1 plants, half the gametes will receive a purple flower allele, which is denoted here with an uppercase P, and the other half a white flower allele denoted by a lowercase p. During pollination among the F1 plants, the gametes unite randomly. An egg with a purple flower allele has an equal chance of being fertilized by a sperm with a purple flower allele or one with a white flower allele. That is, an uppercase P egg may fuse with an uppercase P sperm or a lowercase p sperm. Because the same is true for an egg with a white flower allele, there are a total of 4 equally likely combinations of sperm and eggs. The diagram at the bottom of the figure in the last slide is called a Punnett square, and it highlights the 4 possible combinations of gametes and the resulting 4 possible offspring in the F2 generation. Each square in that Punnett square represents an equally probable product of fertilization. For example, the box in the upper right corner of the Punnett square shows the genetic combination resulting from a lowercase p sperm fertilizing an uppercase p egg. So according to the Punnett square, what will be the physical appearance of these F2 offspring? 1/4 of the plants have 2 alleles specifying purple flowers. That means uppercase P, uppercase P. Clearly these plants will have purple flowers. 1/2, that means 2/4 of the F2 offspring have inherited 1 allele for purple flowers and 1 allele for white flowers, which means an uppercase P and a lowercase p, like the F1 plants, these plants will also have purple flowers, the dominant trait. Finally, 1/4 of the F2 plants have inherited two alleles specifying white flowers. That means lowercase p, and will express this recessive trait. Thus, Mendel's model accounts for the 3:1 ratio that he observed in the F2 generation. Geneticists differentiate between an organism's observed traits, which are called the phenotype, such as the purple or the white flowers, and its genetic makeup, called its genotype, such as whether it's two uppercase P's or two lowercase p's, or an uppercase P and a lowercase p. Mendel studied 7 characters in pea plants. Each character had 2 alternate traits, and Mendel concluded that all these 7 characters had the same type of inheritance pattern. And what was that pattern? That one of the 2 parental traits disappeared in the F1 generation. That means the recessive trait disappeared because it was dominated by the dominant trait, only to reappear in 1/4 of the F2 offspring. And this underlying mechanism is explained by Mendel's law of segregation, which is that the pairs of alleles segregate during gamete formation, and the fusion of gametes at fertilization creates allele pairs again. Now, research has shown that the law of segregation applies to all sexually reproducing organisms, including human beings. Mendel's law of segregation, the relationship between alleles and homologous chromosomes. The diagram in the next slide in figure 9.7 shows a pair of homologous chromosomes, which are chromosomes that carry alleles of the same genes. So you will see a chromosome shown in red color and the other homologue in blue color, and each of the homologous chromosomes, they carry 3 gene loci. What is a locus? A gene locus is a specific location of a gene along a chromosome. That means exactly the point at which the gene is present. So alleles, which are alternative versions of a gene, reside at the same locus on homologous chromosomes. However, the 2 homologous chromosomes may bear either identical alleles or different ones at any one locus. This is figure 9.7 showing the relationship between alleles and homologous chromosomes. You see, one homologue is in red and the other one is in blue, and each homologous chromosome has 3 gene loci. The matching colors of the corresponding gene loci highlight the fact that homologous chromosomes carry alleles for the same genes at the same positions along their lengths. Mendel's second law, the law of independent assortment. So Mendel reached his law of segregation by following the inheritance of one character through 2 generations. So a cross between 2 individuals that are heterozygous for one character is called a monohybrid cross. Mono means one. For example, when Mendel was looking at just one character, let's say plant height, he crossed a tall plant versus a short plant. So these are 2 plants that are heterozygous for the same character, which is plant height. Now, what would be the result from a dihybrid cross? Di means 2. That means a cross between 2 organisms that are heterozygous for 2 characters being followed. So let us take a plant that is tall and let's say has purple flowers, and it is crossed to a plant that is short in height and has white flowers. So there are 2 characters being considered, plant height and flower color, and the 2 plants show differences in both these characters. The question was answered when Mendel crossed the F1 plants with each other to produce the F2. There were 2 alternative hypotheses for gene assortment in a dihybrid cross. First hypothesis is if the assortment was dependent, and second hypothesis is if the assortment was independent. So let's say this is a case of dependent assortment. That means if the genes for 2 characters were inherited together, then the F1 hybrids would produce only the same 2 kinds of gametes that they received from their parents, and the F2 generation would show a 3:1 phenotypic ratio. If you didn't understand what this text meant, don't worry. We will see this in one of the subsequent slides where it is shown in a diagram, in a figure. So then it becomes easier to understand. The second hypothesis is for independent assortment. That means the two seed characters sorted independently. Then what happens? You see, there are these three things that will happen. First is the F1 generation would produce 4 gamete genotypes in equal quantities, and the F2 generation would have 9 genotypes producing 4 different phenotypes in a ratio of 9 to 3 to 3 to 1. And the Punnett square in the next slide would, reveals what a dihybrid cross produces, okay, if it's equal to 2 monohybrid crosses occurring simultaneously or not. This is figure 9.8 illustrating the testing of alternative hypothesis for gene assortment in a dihybrid cross. Only the hypothesis of independent assortment was supported by Mendel's data. So Mendel crossed homozygous plants having round yellow seeds. So genotype is 2 uppercase R's and 2 uppercase Y's, with plants having wrinkled green seeds. The genotype is 2 lowercase R's and 2 lowercase Y's. So it is clear that the round shape is dominant over the wrinkled shape, and the yellow seed color is dominant over the green seed color. So as shown in figure 9.8 here in this slide, the union of an uppercase R and uppercase Y and lowercase r and lowercase y gametes from the P generation yielded hybrids heterozygous for both characters. That means an uppercase and lowercase r and an uppercase and lowercase y. That is dihybrids. As we would expect, all of these offspring in the F1 generation had round yellow seeds. That means the two dominant traits. But were the two characters transmitted from the parents to offspring as a package, or was each character inherited independently of the other? Now, if you look at the Punnett square, okay, it shows that all possible combinations of alleles that can result in the F2 generation were made, okay, and all possible combinations that could result from the union of 4 kinds of sperms with 4 kinds of eggs were produced. If you study the Punnett square, you'll see that it predicts 9 different genotypes in the F2 generation. These 9 genotypes will produce 4 different phenotypes in a ratio of 9 to 3 to 3 to 1. So the Punnett square here also reveals that a dihybrid cross is equivalent to 2 monohybrid crosses occurring simultaneously. So from the 9 to 3 to 3 to 1 ratio, we can see that there are 12 plants in the F2 generation with round seeds compared to 4 with wrinkled seeds, and 12 yellow seeded plants compared to 4 green seeded ones. These 12 to 4 ratios each reduce to 3 to 1, which is the F2 ratio for the monohybrid cross. So Mendel tried his 7p characters in various dihybrid combinations and always observed a 9 to 3 to 3 to 1 ratio. That means 2 simultaneous 3 to 1 ratios of phenotypes in the F2 generation. These results supported the hypothesis that each pair of alleles segregates independently of the other pairs of alleles during gamete formation. In other words, the inheritance of one character has no effect on the inheritance of another. This is called Mendel's law of independent assortment, and it is seen in the inheritance of characters in dogs like the Labrador retrievers. Another application of the law of independent assortment can be examined in this dog breeding experiment shown here on this slide. The inheritance of two characters in Labrador retrievers is controlled by separate genes: black versus chocolate coat color and normal vision versus the eye disorder PRA. Black labs have at least one copy of an allele called an uppercase B. The uppercase B allele is dominant to a lowercase b, so only the coats of dogs with genotype lowercase b are chocolate in color. The allele that causes PRA, called lowercase n, is recessive to allele uppercase N, which is necessary for normal vision. Thus, only dogs of genotype lowercase n become blind from PRA. If you mate two doubly heterozygous labs, that means an uppercase b, lowercase b, uppercase n, lowercase n, which is bottom of figure 9.9 here, the phenotypic ratio of the offspring F2 is 9 to 3 to 3 to 1. These results resemble the F2 results shown in the previous slide, demonstrating that the coat color and PRA genes are inherited independently. Using a test cross to determine an unknown genotype. What is a test cross? It is a mating between an individual of a dominant phenotype but unknown genotype. That means, phenotype could be because of homozygous or heterozygous genotypes. And when you mate such an individual with a dominant phenotype to a homozygous recessive individual, that is known as a test cross. And we will see, learn about this in details in the next slide. This slide shows a Labrador Retriever test cross. To determine the genotype of a black lab, it can be crossed with a chocolate lab, which is a homozygous recessive lowercase b, lowercase b. If all the offsprings are black, the black parent most likely had the genotype uppercase B, uppercase B. If any of the offspring are chocolate, the black parent must be heterozygous uppercase B, lowercase b. The rules of probability. Mendel was an excellent scientist. He meticulously kept records of whatever his observations were, and then his background in mathematics, particularly in statistics, helped him do the statistics, the data analysis, the statistical part, very well. So he understood that genetic crosses, they are obeying the rules of probability. Probability means those are the same rules that we see when we toss a coin or we roll a dice or we are drawing cards. So the first rule, the rule of multiplication in probability, states that the probability of a compound event, that means a complicated event, is the product of the separate probabilities of the independent events. Segregation of alleles and fertilization as chance events. When heterozygotes, uppercase B and lowercase b, form gametes, segregation of alleles during sperm and egg formation is like two separately tossed coins, that is two independent events. You can see in the F1 genotypes, both of these are heterozygous individuals with an uppercase B and a small case b, and you see how the formation of eggs and sperm are like tossing coins, independent of each other. When geneticists have to figure out the transmission of a particular character in a family, they start with analyzing the results of matings that have occurred in the family. So they collect as much information as possible about the family's history of a particular trait, a particular character, or a particular disease. And then the geneticist assembles this information into a kind of family tree, which is called a pedigree. And Mendel's laws help us to deduce the genotypes for most of the people in a pedigree. This figure shows a pedigree tracing the incidence of freckles versus no freckles, a trait which is believed to be controlled by a single gene. The letter F stands for the dominant allele, in uppercase F for freckles, and a lowercase f symbolizes the recessive allele for no freckles. What is the relationship between phenotypes and genotypes? The dominant phenotype, freckled, results from having either one or two dominant alleles, that means an uppercase F in the genotype uppercase F or uppercase F, lowercase f. If both alleles are recessive, that means lowercase f, the phenotype is recessive, no freckles in this case. In the pedigree, a square represents a living male, a circle represents a living female, and usually the square or the circle, when it's filled, then it indicates that a person has the trait, while an empty square or a circle represents the person does not have a trait, although it is shown opposite in this figure. But let's go to the next slide to see the common symbols and lines that are used in pedigree analysis. If you see here in this figure, the blackened or the darkened circle and square, it represents female or male who expresses the trait, while the non-filled or the not blackened circle and square, these represent normal females and males. Please keep in mind that the word dominant in genetics does not mean that a dominant phenotype is either normal or more common than a recessive phenotype. Instead, dominance means that a heterozygous genotype, in this pedigree an uppercase F and a lowercase f, results in the dominant phenotype. Traits that are most common in nature are called wild type. Wild type has nothing to do with dominance. A recessive trait may in fact be more common in the population than a dominant one. For example, the absence of freckles, which is a lowercase f, is more common than their presence, which is an uppercase f and a lowercase f, or uppercase f, that means two uppercase f's.